Hello, everybody. Okay, today we have a very interesting topic. Okay, so today we are actually moving towards pathways. Several coffee breaks before, we were talking about just basic TRANSFAC, how to apply it to search for transcription factor binding sites, to study transcription factors. Today we have next step from transcription factors to the pathways. And the title of the topic today and the presentation today is somewhat don't do pathway analysis without TRANSFAC. Of course, you can do everything you want, but I want to put this in such a special question that when you do your pathway analysis, you may miss a lot when you don't consider the TRANSFAC point of view. And what do I mean? So actually, like on this picture which you see here, the pathways which are acting in the cells and the analysis of pathways which you are doing is actually based on those on the information which is collected from the literature about those canonical pathways, about the connection between different proteins, different genes, and those connections, those reactions, those interactions are taken from literature. They are taken from different studies, experimental evidences, and we actually are based on that corpus of knowledge which was collected from many years from the scientific literature about those protein-protein interactions, about the reactions which are happening in cells. But something which is really very important in those pathways and which is often missing in those pathways, which we use to study our data or to map our data on pathways, actually the interactions between transcription factors and their binding sites. You may argue that there are a lot of techniques and experimental techniques like ChIP-seq and others which actually discover those interactions. So discover those transcription factor binding sites. So we can add to this pathway those connections which are taken from the different experiments about which transcription factor is binding to which site in which gene and how it is regulating those genes. But I can argue that those connections, those interactions are far from being complete. If you dig through all the literature, which we do actually in TRANSFAC, you may be just having only 1% of the knowledge and only 1% of the real transcription factor binding sites, which are actually happening in the cell. So you still have to run predictions of those binding site predictions, which you can do with TRANSFAC or with other tools, but you still have to predict those interactions, those multiple interactions to complete the pathways you are working with, and not to miss the mechanism of diseases you are studying, or really to find what you need. And just to show it in one example, I will show today one set of data which we analyzed here with our tools, which is taken from this pretty recent paper in this year, where the authors of this paper, they took actually fibroblasts, and they were treating them with various things, and that's actually a complicated study they have performed here, but the main target was fibrosis. Actually, and the fibrosis is a very bad problem, which is happening in many tissues in human body, in different diseases, and it leads to various pathological complications. And the main factor, it's known that the main factor for fibrosis is TGFβ. So those cells were treated with TGFβ and with other, well, actually, as I said, the study was more complicated, but one part of it, they were just treated with TGFβ, and actually these cells were transformed and they lead to the fibrosis. And of course, the main question which many people asking, if you have such a treatment, or you have some changes in the cell which happening during, for instance, disease, then they lead to many different processes in the cell, which in fact kind of downstream from that event. So what you see when you do the gene expression study, for instance, in the cells after treating by TGF-beta, then you see a lot of genes changing the expression, and then these are effects, so sort of downstream effect of what happening with TGF-beta after treating the cells by TGF-beta. And that's like those effects which are happening afterwards. But if you don't know what happens, what was the reason of those effects, like you don't know in many diseases, you actually have to look upstream rather than downstream. You have to look not what is happening after, those things are changed, but rather what was the reason of those changes. So in this case, in this particular experiment, actually we know the reason of those all changes which were observed. It's a TGF-beta stimulation, right? So which means if I see the data, if I take this data and I extract, for instance, a set of upregulated genes, and then I would like to have such tool which will give me the reason of those changes. All right, let's have a look. So for that, I extracted this data. So I have it here. Here is actually the original data. This is RNA-seq, which was done on 3 samples of fibroblasts, which was a control, and there were 3 samples which were treated by TGF-beta. And you see here, these are famous RNA-seq counts. Maybe I can make it a bit bigger so you can see it more clearly. So which can kind of a proxy of gene expression measurements. And so then basically you can see that for some genes, so in the control situation, the counts were a bit higher than after treating by TGF-beta, or other way around, a bit high, in the TGF-beta. After TGF-beta treatment, the counts become higher. So basically, genes are either upregulated or downregulated. So this kind of data can be easily analyzed with various tools in our platform, for instance, which I showed before, or you can find many other tools. You can run it with R or with Python, many different methods to analyze such type of data and to reveal differentially expressed genes. So actually, what you get, and which I actually did, let me show you here. I extracted this set of upregulated genes, and there are actually 345 upregulated genes were revealed after TGF-beta stimulation of these fibroblasts. And you see they're sorted by log-fold change. So the highest log-fold change are for those genes. And we have here the p-value and adjusted p-value. So what I did here to select these upregulated genes, I actually put a cutoff for log-fold change higher than 0.5. It's like a reasonable, very low cutoff. And the p-value cutoff, I used 0.01, 1%. So that's a kind of a standard filtering criteria to reveal these upregulated genes. So you remember we have 345 upregulated genes. So now I have these upregulated genes, and I need a tool which will give me answer. That was a TGF-beta stimulation, right? That's what I want. So that again, so what actually I want, if I wouldn't know that it was a TGF-beta stimulation, for instance, if there is a case of disease. So I have just, I observe a disease, tissue and the control, it's kind of non-disease, and I want to compare it. I get upregulated genes, and I want to know, I want to have such a tool which give me an answer that that the reason for those changes, the cause was something which I want to then target, maybe with a drug and so forth. So I want to have a drug target which is a causative. I don't want to be kind of targets which are effects of the causalities. I want the causative track target. All right, so then what I do, I just go to standard pathway analysis tools in order to find a pathway which would tell me that what was the reason. All right, so let me then start again this presentation. So, and there are a number of pathway analysis tools, and actually, for instance, this is a very famous pathway database, REACTOME, very good database. They have a lot of pathways, and they have very nice tools, very user-friendly interface. You put there these 345 genes, and so unfortunately, the title, it's very small letters here. So maybe I can increase this somehow that you see the better. But actually, here's a list of pathways which this tool found. And there is a smooth muscle contraction. There is extracellular matrix interactions. So the pathway I'm actually here showing on the screen is a extracellular matrix organization pathway, which has 20 genes, actually proteins, from my list of genes, my list of 345 proteins. 20 of them were actually mapped to this pathway, and there is a significant enrichment. So the p-value here is 10 and minus 4, 3 and minus 4. So it's a significant enrichment of that pathway. Definitely this pathway is triggered by this event. But as I said, this is an effect of treatment. Many pathways are triggered, many processes in the cells start acting, but there is no TGF pathway in the list. So maybe TGF pathway is not originally in the list. No, it was there, but it was not found. Okay, maybe try another tool. Here is, of course, KEGG database. It's a very big database and very efficient and very nice interface. So here again, I run this 345 genes, and I see a big list of pathways like metabolic pathway, cytoskeleton and muscle cells. Again, by the way, muscle cells, motor proteins, and many others. For instance, here, there is cytoskeleton and muscle cells. And this pathway, you see here, those red proteins, they are from my list. So these are upregulated genes in my experiment. And actually, it's giving a little bit of clue what's going on in those cells. In fact, TGF-beta treatment of these cells leads to activations of actins and other extracellular matrix, which actually, in fact, leads to the fibrosis. So the secondary effect is the final effect of the treatment is fibrosis, and that's what we see here when we run, for instance, KEGG. But again, we don't see the reason. We don't see the TGF-beta in the list. So if then I wouldn't know what was the reason, I would try to maybe treat these secondary effects, like, and that's usual. People do. They, instead of treating the reason, they would treat the effects. So, okay, so then maybe we try another one. For instance, there is a famous database called PANTHER DB. Again, I'm running this set of genes, and it's very restrictive, though, this database. We got only 3 pathways, and among them, for instance, there's one with a very high p-value, a very low p-value, actually, integrin signaling pathway, which you see on this screen, was significantly enriched. Again, no TGF-beta. All right. Again, there is another database in the Max Planck Institute for Molecular Genetics in Berlin. They made an effort to integrate many different pathways in one tool. You have here REACTOME pathways, NET pathways, human-seq pathway, BioCarta pathways, many different pathways, those canonical pathways. So you cannot say there is no TGF-beta pathway in that collection. There is a TGF-beta there, but it was not found by enrichment, so you don't see it here in the list. All right, so why it's happening? Let's and maybe as you know, of course, I'm showing here that just standard pathway enrichment wouldn't show you the origin of that changes, and probably you think, aha, maybe your tool will find it. Yes, it will, although it's not so easy, I would say, because actually, if to be the system is quite complicated. So those fibroblasts were treated with TGFβ over 48 hours. So actually quite long. And then during these hours, a lot of effects, secondary effects happening, which mask the original reason. So other type of data, when you see the very quick effect and you do the measurements only a few hours after effect, to find the original reason is either easier, but I choose this example to show that even in such a noisy data, you can find the original signal. All right, so, but how? Again, as I said, the main actually reason is that those pathways we are analyzing are not complete. And why they are not complete? So because of simple things, so I will just drop everything on this slide just to show you that when you consider cell, for instance, of course, the system is even more complicated. There are many different cells interacting to each other. But when you just consider one cell, and this cell is has a nucleus, cytoplasm, the membrane, and there is some receptors on the membrane. For instance, for TGFβ, there is a TGFβ receptor, and TGFβ ligand goes into the receptor, and this receptor is activated, and it actually transfers the signal down to the nucleus, and this is called signal transduction pathways or network. So basically, there are adapters and secondary adapters, and there are kinases, then activating transcription factors, and then activated transcription factors, they start binding to the target genes and those target sites, and they activate expression or repress expression of a certain set of genes. And then those certain genes actually express proteins, and that those pathways we actually, and those proteins are involved in those pathways which we actually have in our databases. We have those pathways which are here. So when you have already proteins and they do something like they do metabolism, or they do, they change the structure of the cell, like those cytoskeleton, these are kind of working pathways, workers. There is some pathways, also pathways in the databases, signal transduction pathways, the kind of regulatory pathways. And there are many of them also in KEGG, in REACTOME. REACTOME is based mainly on those signal transduction pathways, but they often just end up with transcription factors. So you see very nicely this mechanism, and you see which transcription factors are activated, but you very rarely see which transcription factor, which target genes are actually activated through this from these transcription factors. And the reason is that they are very variable. Depending on the cell type, depending on the tissue, the set of target genes can vary. So although this part of pathway may be very stable, like TGF pathway, receptor and all this set of adapters and everything down to the transcription factor level is quite stable in different types of cells. But which kind of genes are activated then is quite different in different types. And these connections you have to predict. So they are not there in the pathways. That's why when you have your set of genes, and you can only see the, you have your set of upregulated genes, you see those genes which already different in the expression, and when you map them on pathway, you actually find those pathways which are acting afterwards. But you don't see the upstream pathways. You don't see what is the reason for that. All right. So, and we call it to search for master regulators in the whole system. And how we do that, we actually have databases in our hands. So you heard about TRANSFAC, and we were talking about that many times. So and again here, It's a database about transcription factors and their target sites. And actually, it's a database which have those positional weight matrices. We will show it again today, which are tools to predict binding sites for transcription factors, to predict these interactions. We have also other two databases. We're not talking about that too much this year, but these are very big databases as well. For instance, the database called TransPath. It's a database on signal transduction network, not just canonical pathways, we have them as well, but it's actually a big network of interactions and reactions in the cell. And we have more than 1 million, we collected more than 1 million reactions from different papers in this database that's describing actually reactions going on in different cell types in human, mouse, and rat. Here we're talking about a human system. So we have a human interactome, sort of. And then we have this another database called Human PSD, which is a database about targets, about proteins which are involved in diseases, which are biomarkers of diseases, which are targets of different drugs. We also have drugs there, and also clinical trials and all this kind of information which helps for medical purposes. All right, so having these all three databases, we do these predictions using these three databases. So again, what we do, we try to predict the reason, right? The cause of those changes which we observe in the gene expression experiment. Okay, so very quickly, I showed it before. First, you have to predict the targets. So once you have, we collected in TransFac those positional weight matrices, for instance, for each transcription factor, and there are more than 10,000 matrices collected in TransFac so far. And then we apply those matrices to predict binding sites in the promoters or enhancers of the target genes. So we also have a system which I was also presenting before, which tries to find combinations of sites, not just individual sites, but combinations of sites for many transcription factors acting together. We call them composite modules. And with that, we actually have a concept inside the system that those transcription factors, they have to cooperate. We're all talking about cooperation. Single doesn't work. Cooperation works. And that's very important also nowadays. So cooperation actually works. And what exactly transcription factors are doing, they actually sense the target region of the gene when they cooperate to each other. So if there is one complex of factors which is cooperating and these factors are active in the cell, they can find the respective set of sites which are in a cluster. We call them composite module. And when there is another set of factors which are active in the cell, they find in the same promoter, they may find a different set of sites. And because of these combinations are enormous, there are so many different combinations possible between different transcription factors, the whole regulations of genes is happening. So that's why in different cell types, in different conditions, the same gene can be targeted for many factors, and the same factor can regulate one set of genes in one condition and another set of genes in another condition. All right, so that's how TransFac is working, predicting transcription factor binding sites. And the algorithm which we have there is called genetic algorithm. So the details we are not talking today, but basically tries to find clusters of binding sites in promoters of target genes. So once we find those target transcription factors which are acting in cooperation together, acting on my target genes, actually acting on those in the promoters of those upstream of those upregulated genes. So we have these 345 upregulated genes in the promoters of those genes. We have those clusters of binding sites for a particular subset of factors which are cooperating together upon TGFβ treatment. So what is next? So these factors are actually activated. And we know the activation of factors is done during the signal transduction pathways, and there might be a certain master regulator. So in this case, we know there was a TGFβ, which leads to this activation of that set of factors. So that's why we have to find this master regulator in this signal transduction network. And that's what is done by the algorithm which I'm talking today. So this is the algorithm which was published pretty many years ago already. First version of that algorithm, we made several several steps of improving this algorithm over the years. But the major idea of that algorithm, just a moment, is very simple. Once you have a set of transcription factors like these blue guys, you want to find which is master regulator can activate them. And you have a network. So then there are algorithms for finding shortest path in the network, basically going through this network backwards. So you see this kinase is activating these factors, so then we can go up. Then there is this kinase, which is activating this kinase, so we're going up, and so forth. So now we are going from all these factors upstream. And then we're coming up to a molecule which can lead through this network to activation of maybe not all, but at least a particular subset of those factors. And then we give we calculate a score for each of that key molecule. We call them key molecules, right? We give a score, and there is some complicated formula, but it's actually very easy. So sorry, there is a action. There is in the above we have actually a number of transcription factors which can be reached from that molecule. And down here, we have a number of factors which are actually in total can be reached from this molecule. And we want to have actually this molecule to be very specific and also quite sensitive. What I mean that it should be possible to reach all of my transcription factors from this molecule. So this M should be high, but also it should be specific. So only this subset of factors should be reached, but not all other factors. So there are some molecules in the cell which are very unspecific. So like they are called hubs. So if they activated, they lead to activation of practically all transcription factor downstream. So those will be downgraded by this score, and those which are specific will be upgraded. And there are some more parameters here of that algorithm. And one important part of that, which we will show online, is actually you see this size of the molecules somewhat different, and these sizes actually they demonstrate the importance of those molecules during the search of these master regulators. How do you know the importance? Actually, we don't know it. For us, without additional knowledge, all the molecules in the cell are kind of equally important. But then, if we have additional knowledge, for instance, if we have data about proteins, proteomics, or phosphoproteomics, we have some additional data in addition to transcriptomics. And from that data, we know that, for instance, this protein and this protein are present in the cell and even activated in these studied conditions. So then we can add this knowledge to this algorithm, and then this algorithm will be running not just in anonymous network, but in a weighted graph. And then those passes through the graph, which goes through those important molecules, will get a higher evidence or higher weight, and then those passes will be more real. Okay, so that's a part of the algorithm which we call context. We using other data, like for instance proteomics data, as additional context in our search. There's one more concept which we have to discuss today is the concept of walking pathways. We have a paper on that, and at the end you will see this reference. Actually, what means walking? It's another word of the term which you may use or may know. It says rewiring. The pathway are rewiring during their work. So, for instance, if there's a normal cell and there is a standard pathway which is acting in the cell, there is a ligand activating receptor, and through the cascade of events, you get a set of transcription factors which are activating their target genes. But in some pathological situations, those genes, the green ones, they start encoding some components of network which start changing the network itself. And at the end, you may even have a new master regulator, so which get kind of hijacking the whole system. What it's often happening in tumor, for instance, the standard pathways, they are rewired to a pathological state. So actually you get a new master regulator which is taking over the whole regulatory role. And the most important characteristic of those pathological master regulators, I would say, are actually those ones which have positive feedback loops. So you see these red arrows and also green arrows. So what does it mean? So if you have activation of that protein, through this cascade of event, it actually working on its own gene. So it will activate expression of its own gene, so positive feedback. And once you have such a positive feedback, it will just increase, because there is a very difficult to get out of such positive feedback. It's like a niche, energetical niche. And then that's why when we analyzing those master regulators, we search for those ones which have these positive feedback loops. And that could be a characteristic of those pathways which are actually acting in the cell. All right. All right, so that's just demonstration of those working pathways that, as I said, is part of this network. Like in this case, this is TNF-alpha network, which is shown here, somewhat, well, different, but looking similar is TGFβ network. So which activate a certain set of, end kinases, and which then activate transcription factors. And what happening, those in different cells, those factors, this whole network, canonical pathways start regulating other type of genes. So the whole pathway is walking away from the original situation. That's why we call it walking pathways. All right. So that's what I wanted to give you as a kind of theory of what we are going to do. But I think it would be interesting for you, of course, to look how to do this in the system. And that's what we are going to do now. Just to show you that we are going to use today a tool called Genome Enhancer. That's one of the latest development which we did of such a quite user-friendly and easy-to-use interface on the web, where you don't need to change too many parameters. And the whole purpose of that tool is to run this master regulator search. So we call it, the whole thing we call upstream analysis. You see, in the beginning I said, usually when you do pathway mapping, you do sort of downstream analysis, what is happening afterwards. But what we are trying to do here is upstream analysis, asking question why that happens, what is the reason. So that's done by this tool, Genome Enhancer, and you will see it in the moment. All right, so then what we will do now, we will see our tool. So this is our platform. If you haven't seen it before, very easy. You just go to our website, you register there, and you get the free account to this platform. So you don't have immediately the connection to all the tools which we have and to all the databases which we have, but as a software, as a tool, a set of tools which you can run, you can get it for like a free account. All right. So what we want to do now, I want to switch this tool to the Genome Enhancer mode. And that's what I mentioned. This is an easy interface to run this upstream analysis. And here I have my, okay, let me see where it is. Yeah, here's my project. And here you see this drag and drop box where you can put your all kind of data. So, and here we have just a set of, we have actually differentially, well, we have original gene expression data. So you remember these were the data. So these were like this whole raw data of more than 20,000 genes and their raw counts. So, okay, so we should basically take this and put it in there. So let me find where they are. So I have this folder. I have my box. So I take this Excel file simply and drag and drop it here. So it's imported. I click on import, it detecting the format. So it detects that this is transcriptomics. And by the way, we as I said, this tool can work with many different omics, and I will show then two types of omics data we are going to do. But here we just use transcriptomics, and you have you can put proteomics, genomics data like VCF files, mutations in genome. You can put epigenomics like ATAC-seq, ChIP-seq data, and also even metabolomics. Five types of omics data can be analyzed here individually or in combinations. All right, and these are gene symbols which I want to import, and it's importing now in the tool. And the type of formats which you can import, just let me show you while importing. As you can see, we are importing transcriptomics, and you can import just a table. You can import cell files or even FASTQ files. For instance, if you have RNA-seq data and you haven't done yet even these read counts, just put FASTQ files and the system will know what to do with it. So, and here there are other omics and other types of files. So it's here. We go then next. And we have this gray box here. So we actually have this data which we have just put in. Right. So this was the data, right? So that's a set of data. So we have to specify. Yeah, let me open it again. Right. So let me just set this transcriptomics data, and okay, right. So we simply take, drag and drop it here. So we have this data here. So and now we click next, and we have to specify target and control. As you see, I have done it already before. I just want to do it for you. So we have this experiment control settings. So we're saying here we have a TGFβ stimulation, and here we have a control. So then we take these samples, which are actually TGFβ stimulation samples. We drag and drop in here, and these are samples which are control samples. We drag and drop in here. So that's all. Then we click next, and here we have to specify a disease. It's not necessary, but if you fibrosis, yeah, that was the disease we are studying here. It just the final step of that upstream analysis is to deal with drugs and everything, this disease. So it's kind of for the upstream analysis itself, the disease is not important, but for the final report, it's interesting which drugs can be used towards those targets which you find. And now here you have to say, okay, it was TGFβ versus control 2. And here you can even specify the tissue, if you know the tissue, right? Here, actually, it was fibroblasts, but from which tissues, I'm not sure. So you can kind of keep it no selected, which means it will take promoters of the genes from the general genes, general gene promoters from ensemble database. If you know the tissue, it will take this information into account while selecting promoters of those genes. And then you simply start the analysis. So, and then it will run for quite some time. So I will actually stop it for now. I break it for now because it takes a lot of resources from the server. So I don't want to interfere with our demonstration. So I would just show you the final result. Well, it opens this as a complete sort of report. As you can see, we have here started treatment versus control. But I want to show you how does it, without many steps in between, just to show the final results. Let me go back to the platform. And in the platform, actually, you can see those results very clearly. So let me go to that place in the tree where those results are located. So first of all, it took this whole set of data and it started analyzing it, and it identifies upregulated genes, right? So it gives the list of upregulated genes. And then it started to analyze the promoters of that upregulated genes as I showed you in my presentation. And it finds composition of those binding sites in the promoters of the genes. And that's what you can see here in these results. So let me open this for you. And by the way, I have one question. Yeah, in between, there is a question. Do we give input as raw counts or the upregulated and downregulated genes separately? Right, you can do both, either way. So what I did now is I put the raw counts, and it analyzes these raw counts by itself, the system, and defines the upregulated genes and downregulated genes according to the parameters which we have in the system. You actually can set those parameters when you start the run, but by default it uses particular log-fold change cutoff and p-value cutoff. If you have your own way of selecting the differentially expressed genes, you can do it before by yourself, and then your input would be just a set of genes which you want to analyze. These are 100 genes which are upregulated. Here they are, just analyze these, and the system will do it like you want. As I said, you can either put FASTQ files, then it will process all the time, all the way towards the counts, and then alignment counts, and then differentially expressed genes. Or you can put directly up the differentially expressed genes, then it skips all these initial steps and proceeds from your set as it is. All right, so here I have this list of genes which are upregulated. And here are promoters of those genes. For instance, you see this red bar is the first exon of HR gene, for instance, or this collagen gene. By the way, collagen here is activated. And here is in front of this gene, there's another gene, by the way, going in different direction, but in front of this gene, there is a promoter. And here you see this number of sites found to be in clusters near the promoter start site. And which cluster of sites it is, you can see it here in such a form. You see this is a set of positional weight matrices which the system selected for you. You know, the TRANSFAC has, for vertebrates, it has 8,000 positional weight matrices. And the program was combining, trying all of these 8,000, and then it selected these 10 matrices together that they give these clusters of sites in this set of promoters, and it discriminates the promoters of the target genes, of the upregulated genes, from the promoters of non-change genes. You see the discrimination here is, well, it's significant, not tremendously good. We'll see better discrimination in a different data set, but it's significant. So there is a signal which shown in those promoters. And these are transcription factors which are combined in those clusters. Okay, what is the next step? You take the system takes these transcription factors, and it start searching this whole network for the master regulators. Well, it's doing many things, but let me just show you the final result. Here is this network. Okay, so you see this is a network. So the blue bars here are the target, are the genes which are upregulated. So you see not all of them, but those which are mostly upregulated. So the blue guys here are the transcription factors, which are found to be in the clusters of sites for these transcription factors were found in the promoters of those genes. And these green guys are the connecting molecules in the network, and the red guys are master regulators. And you see already here this complex, which was found by the system, complex of molecules, which includes TGFβ itself, with TGFβ receptors and other adapter molecules. So such complex exist, and you see these phosphorylation events which are happening in this complex. So it's all these complex, by the way, is present in our TransPass database. And here we have this reaction which was found in this network of our database, which is activated by this complex, and basically it activates this ATF2, which activates in other molecules until it activates all this set of transcription factors working on that set of genes which we see. So we have, of course, not only this master regulator here, but we have other master regulators found by the system. And as I said, this is 48 hours after stimulation, and during the stimulation, there is a lot of secondary effects happening in cells. So you see other activated and secondary master regulator molecules. So you see this moving pathway in work. So this working pathway. So if originally the original network which was activated was started from TGFβ complex, but then many other genes were activated which start playing their activation role, a master regulator role in this network, and you see this network already rewiring and the cells moving and acting. But what is important, you will be able to retrieve the original signal. All right, so that just to show you, like, the simple mapping, often it always works. It gives you a real pathways which are activated, but these are downstream effects. And to find upstream, to find the cause of those effects, you have to actually apply TRANSFAC, because then you know which transcription factors are activating my genes, and then you go upstream from those transcription factors, you kind of reverse the time back, and then you go to that master regulator molecules, and hopefully these are the causative molecules, and this might be your targets. Okay, so just in the end, I would like to show you one example of that other set of genes. Let me open that. Yes, here. When I put two omics types, and I mentioned in my talk that we have this algorithm of finding master regulators, and in that algorithm we have those molecules which are bigger and smaller. So we can use the context. And for instance, if you have proteomics data, you can use this as a context. And here I want to present one set of data, which is from this paper. It's a pretty old paper, but here it's a completely different system. And these are osteosarcoma. And this osteosarcoma is driven by MYC. And they activated this MYC in the cell line, and they did measurement of RNA-seq. They did actually expression measurements, gene expression, and they did also proteomics studies. So that's why you have here transcriptomics, the same as in before, but in addition, you have proteomics, basically the list of proteins and the abundances, the changes actually in abundances after treatment by MYC, after activating MYC, actually, there was internal activation of MYC. So, and then I put these two types of data in this system. And here I want to show you the result. So here, if you go in that type of data, the initial setup is very similar. I put expression data in the MYC activation and in control. In addition, simply I put here the proteomics data. And that's the general way how to put data inside in the Genome Enhancer. If you have other type of data, you simply put it in this box. Transcriptomics with proteomics all together. And the system knows, okay, this is transcriptomics, this is proteomics. It starts analyzing it, start comparing it. So it's doing a lot of things here. So you see a very colorful report. And one thing which is interesting here is it actually compares different types of omics data. For instance, here, the proteome versus transcriptome, it finds they are correlating to each other, which is a very good sign. These two types of events are kind of co-occurring. So there's not always the case when transcriptomics and proteomics are correlating. In this case, they were correlating. And then you see this, in this case, discrimination is much better. The signal upon MYC, MYC is actually transcription factor by itself. So a lot is happening in the promoters of those genes, and the discrimination is much better. And the final result is similar. So you have this network and those master regulators. And by the way, the MYC, you see the MYC here was found also as a master regulator here by the system automatically. And then what I wanted to show to you at the end, it's actually very practical. So at the end, this tool also gives you a list of drugs which can be used, or either known to be used for that particular disease, either they are already approved or they are in clinical trials. And you see here the drug name and the target, which we found here as a master regulator. So that's the final step, which we do in our tool, like I showed here. You know, when we find master regulator using human PSD, we predict which target, we use it as a target, and we predict which drugs can be used for this particular case. All right. So of course, just generally in osteosarcoma, that's not not something new that MYC is important target. But what is actually, there are two things. First, in different patient, different targets can have a different activity, and that system can prioritize which drugs are better for a different patient. And the second thing, it can do repurposing. So it checks which other drugs in the system and which other compounds in the system can work on the same targets we found. But that's another story. We will have another coffee break for that, I hope, about drug targets and how to find a good drug. Here, I just wanted to show it to you that don't do pathway without TRANSFAC. That's the main message I wanted to tell you today. And that's basically it for today. So I think we have enough time for your questions. Again, this is a geneXplain platform. And in this platform, we used one tool, which is called Genome Enhancer. But basically, this Genome Enhancer is one big pipeline, which is based on that tool. And you can run the whole pipeline as I showed today, or you can run individual tools here in the platform. And by the way, when you have your free account, you can see those tools. For instance, if you go to in this platform, you go to this method section. Well, first of all, let's go to the home, and you see those different sets of tools we have. They're split by type of data you're analyzing. You can analyze RNA-seq data, microarray data as well, chip-seq data, as we discussed, and ATAC-seq data. Also genomic variants, like you have VCF file and you want to know which mutations are affecting your target genes, or which mutations are affecting transcription factor binding sites. And you can do it here. And once you click, you have here just tabs. So you can load your data, you can pre-process it, you have can detect differentially expressed genes. You can simply discover functional enrichment. Similar way like you do with KEGG or with REACTOME. Here you map your genes on pathways. That's important to know, as I said, to do this downstream analysis to see what's happening in your system afterwards. That's important step, and you can do it here. You can do this network analysis, this finding master regulators separate. No, I showed you like in the whole pipeline, you start from the very beginning, and this pipeline is doing this all automatic, but you can do it separate promoter analysis, separate master regulator. So these are the pipelines, and in those pipelines, you actually can even see those pipelines. Yeah, I'm talking just because I'm waiting for questions. Just put your questions, and I'll stop talking. But let me talk. So you see here, there's these are those workflows, and you can run them. They already put together. So like you do the first step. You take your data, do the regulator search, and then you convert the data, you prepare it in a different format. So these workflows are already there, but you can actually edit your workflow, and that's a whole story, how to add new steps in the workflow, how to change parameters of workflow. That's quite easy. It's you have here the user-friendly interface. When you open your workflow and you click and you change the workflow parameters, you add here new tools and so on. So all here on the screen. So these are workflows. Separate, if still workflows are not enough for you to do your work, you can go directly to each individual method. And here we have a set of methods in the tree, and they're all kind of methods, starting from just data manipulation, where you can convert tables, intersect tables, annotate tables. You can also work with tracks, so those sites can be seen on the genome browser. And then so then you have specific tools, like for site analysis. You have tools to run the TRANSFAC site search. You have tool for to find the composite modules, those composition of sites. There are tools for molecular network analysis. The tool which I showed in the pipeline to search for master regulators, which is it's actually using this the so-called regulator search. And this is individual tool. You can change here different network which you are actually using. So I showed you we using TransPass as a network in this tool. But if you don't have TransPass, for instance, you don't have license for TransPass, but you can use REACTOME instead. or recon. Recon, it's a metabolic network, a reconstruction of human metabolic network. So that's those different hubs, we call them hubs, you can use to find the master regulators. So these are individual tools. And here we have, by the way, also statistical tools. Just to do your standard statistical work with your data, like you do your heat map of your data, or you do the PCA plot, or even train your SVM, support vector machine, or train random forest. So you have some element of machine learning here in the system. All right. So okay, so I have a question. Is it possible to analyze the structure of promoter, like which sequence is necessary and which is dispensable? Okay, using ataxic data. Right, that's a very good question. Yes, we have here a toolbox, a set of tools for it's called ChIP-seq, but the approach is very similar. If you click here, there are a number of tools to pre-process your ataxic data. So, for instance, you first load it as a like bed file, for instance, already mapped. So you have regions in genome, or maybe you have your FASTQ files. Then we have tools to align them. And then once you align them, you can do the peak calling. We have this Max algorithm for peak calling, where you can put your aligned data here, those BAM files, and do the alignment. And then you get your peaks, right? And then in these tools here, you see analyze ChIP-seq peaks. Once you have your peaks, so you can identify enriched TFBS in these, well, they're called in ChIP-seq peaks, but the same is in the ataxic peaks. You just need peaks, regions in genome, and you put them here. You define the species. We are working in different types of species, different genomes, and it will analyze those parts of promoters areas where you see the peak. Well, and of course you have to either put all the peaks together and it will analyze all of them, or you can intersect with the promoters of the upregulated genes, if you have RNA-seq and ATAC-seq together. And by the way, this intersection is done automatically if you use Genome Enhancer and put there simply RNA-seq data and ATAC-seq data. And this pipeline will first take promoters of differentially expressed genes, extending actually not very short promoters, but really long promoters, like up to 5,000 kb, 5 kb upstream from TSS. And then it will search, it will look if your ataxic peaks are inside those regions. And if they are, it will take those regions only, as you said, it will exclude the regions, other regions from the analysis, but just focus on those which are covered by the ataxic. So that's all done automatically in this Genome Enhancer pipeline. If you don't want to do it in one pipeline, but step by step, as I said, there are tools to work with promoters, with these peaks together, to overlap them, to concatenate them, and so on. So this number of tools working with join tracks, intersect tracks. And then once you have your tracks, you put them here and analyze the enrichment of transcription factor binding sites. Thanks a lot. Thanks a lot. Yeah, thank you for your interest. So in the meantime, I can show you something, since you were asking about this ataxic data. So for instance, if we have these ataxic peaks, this is classical BD file, so you know what it is, right? So you have basically from to chromosome number, and there's some values which are associated with the peak. So you can take this file and just upload it here, clicking here, computer, and you go to the folder where your peaks are, and we take this BD file. Okay, let's take this one. And it automatically recognizes it's a BED format. Now you have to know which is a build. So in which species, of course, because there is not such an, often there in BED file, we don't have this information. So then in this case, I know that it was from build HG19. So not the latest build HG38, but HG19 of human. So you have to select that. And then you simply start. So it's loading into the system and it's automatically opening it in the genome browser. So you see your peaks. All right, so you can zoom it in, zoom out. So now it's just a chromosome and the peak. So you can actually put here more information from the genome to know which genes these are and so on. So you can put more tracks. For instance, track with genes, track with variations. You just drag and drop them here. Maybe karyotype and so on. So you can put these more peaks here. More information. So then you know here, for instance, which variations there are. Well, it's still reading the genes, so it's quite a big set. But once you have it, you know, yeah, you see in which genes these peaks are located. So for instance, this is located in that gene, in the beginning of that gene. So it's a good candidate to analyze it. So and that's why you have those peaks. And as I said, you can apply it to search for the predicting transcription factor binding sites, and then next step would be for the master regulators. Okay, I think we can finish for today, and thanks a lot for your questions and for your interest. And come back to these coffee breaks next time. Look at our website for the next coffee break. Prepare your questions and welcome back. Okay, see you and have a nice day. Goodbye.